Alright, so in this video, we're going to be talking about a little bit about sticky ends and blunt ends and how to make and how to do cloning, mainly cloning. So we have different cleavage sites, places where the DNA is cut. So this cut would be sticky end or staggered end, and this cut would be a blunt end, and this also is a blunt end. That's all you need to know about this. And then if you want to join the sticky ends, this part will do hydrogen bonding, and then you use DNA ligase with the help of ATP to bind it. It's easier to do these than these because they come together better because of the hydrogen bonding. There's no hydrogen bonding here, and the ligase just has to attach them to. Alright, so we'll go through this real quick. A DNA sample is cut with a restriction enzyme. If the whole genomic DNA is used, millions of different restriction fragments may be generated. So if you have 3 billion bases in the human genome and you cut it with a restriction enzyme that's recognized it every 3,000, then you have a million. But if you're doing like a cDNA, so if you have all the cDNA of a specific tissue type, then you might get 10 to 100,000 bases of 10 to 100,000 fragments, restriction fragments. Note that many restriction enzymes produce restriction that do not have overhangs. So like this one does not have an overhang. So a few bases now have complements, have cohesive ends or sticky ends. So some have sticky ends. So this would be a sticky end. That would be a sticky end. A vector is cut with the same restriction enzyme, but the vector contains a single site for the restriction enzyme. So like in the human genome, we said it would have 1,000,000 sites where it would cut, no, 1,000 sites where it would, actually, yeah, 1,000,000, it would have 1,000,000 sites where it would cut, but in a in a vector or a plasmid, vector could be a plasmid, it could be a bacteriophage, and different things. We'll see that slide later on. And in a plasmid, if it's circular, it'll make a single cut, and then it'll go from circular to linear. If it's a phage, then if you cut, you have two arms. So if it's like linear already, and you make a cut, you get 2 fragments. Restricted vector and sample are mixed and allowed to anneal. Annealing is that the overhangs will find complements. So if you take excess of the vector DNA and then you have more of the vector, the DNA that you want to incorporate into the vector is going to go into the vector. So you have the vector, and then this is the DNA you want it to go in there. So you take a lot more of this, then it'll go in here instead of the piece that came out of here going back into it again. So by taking excess of the vector DNA, we aim for preferential annealing of the sample DNA fragment to the vector fragments. The annealed overhangs are sealed by DNA ligase. The sample DNA incorporated we call insert DNA. So this part over here is called the insert DNA. In most cases, represents single restriction from the original sample. So you just use one restriction enzyme. The ligated DNA mixture is transfected to bacteria. So you heat shock the bacteria or do other methods to get the plasmid that's containing the gene of interest in the vector. So like you poke holes into the bacterial cell membrane using heat shock or salt or other stressors for the bacteria, so it incorporate, so it gets holes and then the DNA gets into the bacteria. So there's lots of bacteria, a large excess of bacteria. Make sure that you don't get two plasmids into one bacteria. And then, so each colony of bacteria is genetically identical. So they have the same exact DNA composition. And so each one of those colonies are grown separately and stocked, and then we have a library, DNA library. So we cut the 3 billion base pairs into a million ones, and then we have 1 million different fragments. So there's 1 million stockpiles that are stored somewhere, and each one of those plates has part of the human genome. So each bacteria has part of the human genome. So then, and then you can take one of those, let's say, one of those bacterial colonies, and then take the plasmid, and then you can sequence the insert. So this is a simple way how the genome is sequenced. So if you're interested in a particular DNA fragment, to see if it goes for a particular fragment, and then you can use a probe to find the particular DNA sequence. So you have to know the sequence, and then you do the probing. And then there's pictures on how you do that. And then you can put that plasmid into a eukaryotic cell and then see if there's expression. So cloning, we'll just go through it with the pictures. So you have the vector, and you have the eukaryotic chromosome. Cut this and this with the same restriction enzyme. You got this over here, and this gets cut up into a whole bunch of pieces. And then you take a large excess of this, so that these go into here rather than the piece that came out of here going back into it. Then you have DNA ligase, does the phosphodiester bonds, and then you put the plasmid containing the gene of interest into a bacterial cell. You do that by poking holes into the membrane. You poke holes using shock and chemicals or salt, and then you let the bacteria grow and they'll multiply. And then this is a map of a vector. So you have the sites for restriction enzymes to cut it. So PSTL would cut here, ECOR1 would cut here, BAMH1 would cut here, SAL1 would cut here, PVU2 would cut here. This is the origin of replication. All vectors have to have an origin of replication, otherwise they wouldn't replicate. And if you can't replicate, you can go from here to here. And so you have the vector. These are all the vectors. And then you have an ampicillin resistant site and a tetracillin resistant site. And we're gonna disrupt the ampicillin resistant site using PSTL1 restriction endonuclease. And then the DNA we want to put in here is this stuff, and we're gonna put that in here, and it's gonna have a huge insertion in here, so the ampicillin resistance is gonna get messed up. So the bacteria that do get this plasmid won't be resistant to ampicillin, but they will be resistant to tetracycline because the tetracycline part is still intact. So, and then you have DNA ligase, it puts it together. And then you have these cells put into E. coli again, and then you grow them. See this one, this cell is lost the resistance to ampicillin. The yellow one is ampicillin resistance, the blue one is tetracycline resistance. So this one also lost the resistance to ampicillin. And then you grow it on tetracycline. So all the cells that don't have any plasmid will die, and all the cells that have some sort of a plasmid will live. See, this one doesn't have our gene of interest, just has a plasmid, so that will still be living here. So then what you do is you take a toothpick and you touch one colony, touch over here, and then touch over here, and then take another isolated colony, touch over here, touch over here, and then you do that with all other dots, and then you let it grow. And then you see that over here everything grew because it's only tetracycline. So you expect everything to go because everything that grew here has tetracycline resistance. And over here you have ampicillin and tetracycline, so only the ones that have the gene of interest in the plasmid will be ampicillin sensitive, and the ones that have ampicillin resistance or the plasmids that did not incorporate the gene of interest. So these ones are the ones that are ampicillin sensitive, and they are the bacterial colonies that have the gene of interest in the plasmid. So this is another bigger picture just showing the same thing talking about before. And cloning. So you have human DNA, you cleave it, the restriction nucleases into many millions of genomic DNA fragments, and then you cut the vectors with the same endonuclease, and they'll combine with them, and then you put the plasmids into bacterial cells, and then you isolate each individual colony, and then you have genomic control library. And then let's say you're looking for a specific sequence from this genomic library. So you grow the bacteria on a plate. You take nitrocellulose paper, put it on the bacteria, so you get some of the cells on the paper, and most of the cells will remain on the gel agar. And then you wash it with the alkali, and the cells get exposed and denatured, and the DNA will be the only thing left over. And then you use a radiolabeled probe, and the probe will bind to the ones that you're looking for, the DNA sequences you're looking for. And you wash everything else off, then only the ones the probe is there. And then you relate this back to these two colonies. So these two colonies, then you know these two bacterial colonies have your sequence, the sequence you're looking for. And this is preparation of a cDNA library. So you take a messenger RNA, which has a poly A tail, so then you can use T's in a string as a primer, and then you use reverse transcriptase, and that's gonna make your DNA RNA hybrid. Then you use RNase, it's gonna cut the RNA, and then using the RNA as primers, you make it into a pure DNA strand. And the difference is between genomic and cDNA library. So you basically cut everything up, the whole genome, and then you have fragments, and then you clone them into bacteria. Over here, you have the genes and the DNA, transcription, get RNA, and then when RNA gets spliced into messenger RNA, then you take the messenger RNA, use reverse transcriptase, and you clone. And then after reverse transcriptase, you take the DNA and you put them into bacterial cells by cloning them in there. So then you have a cDNA clone library and a genomic DNA clones in the genomic library. And some comparisons. Source of the DNA, genomic library, chromosomal DNA, source of DNA, mRNA, cDNA. And then we just read this. And you can just read this too. It's just a chart. So this is this one I'll do in next video.